A research vessel in the middle of the Indian Ocean. No moon, engine off. Below the hull, dark vertical shapes in the water, motionless, like submerged tree trunks. They're not tree trunks, they're sperm whales. 7 animals, 15 meters each, standing upright in the water, not moving, heads pointed toward the surface, tails hanging in the darkness below. The acoustic transmitters glued to their backs were supposed to record echolocation clicks. What they recorded was silence. 10 to 15 minutes of nothing, no clicks, no movement, no breathing. The animals had simply shut down. The boat drifted into one of them, hull against skin, 15 meters of animal, and the whale didn't react. Seconds passed. Then one woke, and the entire group fired downward at once, as if an alarm had gone off inside the water. If a 50-ton predator risks death for 10 minutes of sleep, what happens to everything else in the ocean? Our descent is about to begin, so if you want to follow this and future documentaries, leave a like and subscribe. We begin. On land, falling asleep is the most unremarkable thing a body does. You lie down, your eyes close. The diaphragm keeps working. The intercostal muscles keep pulling the ribs apart and letting them fall. The brainstem keeps firing signals to inhale and exhale, and none of it requires your participation. You are gone, and the machinery runs without you. Breathing is involuntary. It happens the way a heart beats, below the floor of awareness, outside the reach of will, constant as gravity. A sleeping dog breathes, a sleeping child breathes, a human in a coma breathes. The architecture is ancient, embedded so deep in the nervous system that destroying the cortex entirely will not stop it. As long as the brainstem holds, the lungs fill and empty, and consciousness has nothing to do with it. Now take that architecture and remove it. A whale does not breathe the way you do, neither does a dolphin, a porpoise, or any other cetacean. Every single intake of air, every expansion of the lungs, is a willed act. The animal decides to surface. The animal decides to open the blowhole. The animal decides to inhale. There is no background process running the operation, no autopilot, no fail-safe that kicks in when attention lapses. The brainstem does not take over. If the mind goes dark, the blowhole stays shut. This means something very specific about unconsciousness. For a creature on land, losing awareness is routine. It happens every night. And the body manages itself. For a cetacean, losing awareness is a sequence that ends in water entering the lungs. Total shutdown of the brain. The kind that happens during deep sleep in terrestrial mammals is not rest. It is drowning. And yet the sperm whale appears to do exactly that. In 2008, a research team from the University of St. Andrews, led by Patrick Miller, was tracking a group of sperm whales in the waters north of Chile. The animals carried acoustic tags glued to their skin, small devices designed to capture echolocation clicks, dive profiles, body orientation. What the tags captured instead was a behavior no one had described before. The whales stopped moving. They hung vertically in the water column, heads pointed upward, tails dangling into the blackness below, and went still. No clicks, no course corrections, no sign of active respiration. 10 to 15 minutes of nothing. When the research vessel drifted into one of the animals, hull pressing against skin, the whale did not react. Then one animal startled, and within seconds the entire group fired downward and disappeared. The tag data suggested something that, for a cetacean, should be impossible. During those vertical intervals, both hemispheres of the brain appeared to enter a resting state simultaneously, not one half at a time, which is how every other studied cetacean sleeps. Both halves, full bilateral shutdown. The animal was, by every available measure, entirely unconscious, floating in open ocean at the surface, with no mechanism to correct its position if something went wrong. The physics that makes this survivable lives inside the whale's head. The spermaceti organ, a massive reservoir of wax and oil filling roughly a third of the animal's total body length, functions as a kind of biological ballast system. The substance inside changes density with temperature. When it cools, it becomes heavier, and the animal sinks. When it warms, it becomes lighter, and the animal rises. The whale regulates this by controlling blood flow through the tissue surrounding the organ. More blood means more warmth, less blood means cooling. Through this mechanism alone, without a single muscular contraction, the animal adjusts its depth. It rises slowly, blowhole drifting toward the surface, and hovers there, the opening centimeters from air. But there is no correction system, no reflex that triggers if the body sinks too far, no automatic gasp if the blowhole goes under. If the current shifts, if the temperature balance tips, if anything at all changes the equilibrium, the animal has no fallback. The brain is off. 7% of the day, roughly 90 minutes in every 24-hour cycle. That is how much a sperm whale sleeps. It is the shortest known sleep duration of any mammal ever recorded. And every second of that sleep is a wager placed against physics, current, and chance, with the entire organism as the stake. Most cetaceans never accepted those odds. Bottlenose dolphins solved the same foundational problem through a different mechanism, one that sounds like a thought experiment until you see the data. Split the brain down the middle, left hemisphere, right hemisphere. Now let one half sink into slow wave rest, deep, restorative, electrically quiet, while the other stays fully online. The sleeping half goes dark. The waking half keeps the animal moving, keeps it surfacing to inhale, keeps one eye scanning the water for threats. The eye connected to the active hemisphere remains open. The eye connected to the resting hemisphere closes. The dolphin swims in slow loops, rising at intervals, navigating, monitoring, with half a mind. After a period, the sides reverse. The rested hemisphere takes over. The tired one descends into slow waves. The open eye switches. The cycle continues back and forth, hour after hour, and the animal never once loses contact with the world entirely. In 2012, a US Navy-funded study pushed this system to its apparent limit. Researchers at the Navy Marine Mammal Program required bottlenose dolphins to detect underwater targets using echolocation, not for an hour, not for a day, but continuously, around the clock, for 15 consecutive days. No pauses, no off periods. Day and night the dolphins pinged and listened, alternating hemispheres, never allowing both sides to rest at once. At the end of 15 days, their detection accuracy had not declined. The performance on day 15 was indistinguishable from day 1. The system did not degrade, it did not fatigue. The divided brain was not an improvised workaround. It was a precision instrument, built to run indefinitely. But there is a window in every dolphin's life when even this instrument is not enough. Newborn bottlenose dolphins do not sleep. Not in reduced amounts, not in fragments. They do not sleep at all. For the first month after birth, the calf moves continuously, 24 hours a day, every day, without interruption. The mother matches it. The two swim in echelon formation, side by side, bodies nearly touching, surfacing together, turning together, never stopping. The calf cannot regulate its own buoyancy with any precision. If it stops moving, it sinks. Its blubber layer, the insulating sheath that adult dolphins rely on for thermal regulation, has not yet formed. If it stops moving, it loses heat, and it is small enough to interest every predator in the vicinity. So it keeps going without pause, for weeks. In every terrestrial mammal studied, newborns require more rest than adults. Kittens sleep 20 hours a day. Human infants sleep 16. The developing brain demands downtime, long stretches of neural consolidation, synaptic pruning, the quiet internal work that builds a functional mind. In the ocean, the opposite is true. The youngest animals are the ones who cannot afford to stop. Research published in 2007 by Oleg Lyman and colleagues at UCLA tracked the eye states of mother dolphins during the neonatal period. Both eyes remained open 93 to 98% of the time throughout the first month. Not one hemisphere resting while the other watched, both hemispheres online, both eye scanning, for weeks on end. In a terrestrial mammal, sleep deprivation of that magnitude would produce cognitive collapse within days. Organ systems would begin to fail. Death would follow. The dolphin sustains it as standard biology. How remains unanswered? The physiological mechanism that allows a bottlenose dolphin mother and her calf to forgo all measurable rest for a month without the catastrophic neural and organ damage observed in every land mammal subjected to similar deprivation has not been identified. The phenomenon is documented, replicated, published. The explanation does not yet exist. What exists is the pattern. In the ocean, every breath is a choice. Every moment of unconsciousness is a moment that choice goes unmade. And every cetacean that has ever lived has had to find its own answer to the question of how to surrender awareness without surrendering the single act that keeps it alive. The sperm whale shuts everything down and trusts the wax in its skull. The dolphin splits its mind in two and runs each half in shifts. The newborn abandons rest entirely and pays a cost the science cannot yet measure. None of these solutions is safe. None is comfortable. None was free. But dolphin can slow down. It can glide, drift, circle lazily near the surface while half its brain rests. It has the luxury of choosing its own pace. For roughly 24 species of shark, that luxury does not exist. Great whites, marcos, hammerheads, these animals extract oxygen by ramming water through their mouths and over their gill slits as they push forward. The mechanism is called ram ventilation, and it has a non-negotiable requirement: movement. Forward motion forces water across the delicate gill filaments where gas exchange occurs. Stop swimming, and the flow stops. The flow stops, and the blood no longer picks up oxygen. Within minutes, the tissues begin to starve. For these species, stillness is not rest. Stillness is suffocation, which makes sleep, in any conventional sense, a mechanical impossibility. Every definition of sleep used in biology includes some version of reduced activity, decreased responsiveness, a body that has settled into stillness or something close to it. These sharks cannot settle. They cannot reduce locomotion below the threshold that keeps water moving across the gills. They do not even possess eyelids to close. The animal is always in motion, always exposed, always driving forward through a medium that will kill it the moment it stops. What scientists observe instead is something they have cautiously termed restful swimming. Intervals during which the body continues its forward trajectory, but portions of the neural architecture appear to dial down. The shark is still moving, the tail still beats, the water still flows, but something behind the eyes may have partially withdrawn. In 2020, Michael Kelly and colleagues at the University of Western Australia captured video of wobbegong sharks and white-tip reef sharks entering brief, repeated episodes of minimal movement. Their bodies angled into the prevailing current so that the water did the work of ventilation, with almost no muscular effort from the animal. The posture was deliberate. The orientation was consistent. The episodes were short, cyclical, and voluntarily entered. But no electroencephalogram has ever confirmed true sleep in an obligate ram-ventilating shark. No one has successfully attached electrodes to a free-swimming great white and recorded the slow-wave signatures that define sleep in mammals or birds. The technology does not yet exist to answer the question cleanly. What exists is observation, an animal that never stops, that appears to partially vacate its own awareness for brief windows, and that resumes full alertness without any measurable transition. The shark is there, and then perhaps it is not entirely there, and then it is back. The body never paused. There is, however, one species that reveals what becomes possible when the constraint is removed. The white-tip reef shark can pump water over its gills through buccal pumping, rhythmic contractions of the mouth and pharynx that force water across the gill surfaces without any need for forward locomotion. This single mechanical difference changes everything. because the white-tip reef shark can stop. And it does. During daylight hours, groups of them pile into caves and overhangs along the reef structure. They lie on the bottom, stacked on top of one another like logs, motionless, eyes open and unblinking, for hours. The water moves through their mouths in slow pulses. The bodies do not move. The scene looks less like a predator's lair than like a storage facility. Rows of grey shapes packed tight in the dark, waiting. When the light begins to drain from the water column, the pile disassembles. One by one, the bodies unpeel from the heap. They slide out of the cave mouths and begin to move along the reef face in loose, unhurried groups. Their heads push into every crack, every crevice, every gap in the coral architecture. What they are searching for is everything that fell asleep when the sun went down. The fish that wedged themselves into holes in the reef for protection. The ones that buried themselves in sand. The ones that trusted the dark to hide them. The white-tip's daytime stillness is not random, it is timed. Its rest period maps precisely onto the hours when its prey is active and alert and impossible to catch in the open. Its hunting period maps precisely onto the hours when that same prey has gone motionless and vulnerable, tucked into shelters that the white-tip's slender body is built to penetrate. The shark's pause is the reef's exposure. One animal's rest is another animal's most dangerous window. In the economy of the reef, sleep is not private. It is a schedule that every species reads and every predator exploits. A coral reef in daylight is a visible, legible place. Hundreds of species occupy defined positions in the architecture. Territorial dam shellfish hovering above their patch of branching coral, surgeonfish grazing along the upper ridges, wrasses picking parasites off larger bodies, parrotfish scraping algae from the limestone surface with fused beak-like teeth. Every animal is in motion, in the open broadcasting its presence through colour and movement and sound. The structure hums. You can read it. The same structure after sunset is unrecognisable. The daytime occupants are gone, not departed, hidden. Every species that was visible an hour earlier has withdrawn into the reef's interior, each one using a different strategy to become invisible. Some wedge themselves into narrow vertical cracks in the coral, bodies compressed until the fins fold flat against the flanks. Some bury themselves in the sand at the reef's base, leaving only a faint outline, a slight irregularity in the grain. Some find pockets in the dead coral where no light reaches, and hover there, motionless, suspended in a posture they would never hold during the day, tilted at odd angles, fins slack, mouths slightly open. None of them close their eyes. Fish have no eyelids to lower. They enter rest with their eyes open, fixed, taking in whatever faint ambient light still filters through the water. They lack a neocortex, the layered neural structure that, in mammals, undergoes the most dramatic changes during sleep. What fish experience when they go still is something researchers have cautiously labelled reversible quiescence, a measurable reduction in movement, a slowed reaction to approaching stimuli, a postural collapse that indicates the body has shifted into a different mode. The animal stops moving. It takes longer to respond when something comes near. Its muscles release tension. The state looks like rest. It follows a circadian pattern. It can be interrupted and resumed. Scientists can quantify it, track it, reproduce it in laboratory settings. They hesitate to call it sleep. But the functional problem is the same, regardless of the label. Every fish in that state is motionless in a place where other things are moving. The reef at night is not empty. It is full of animals for whom the darkness is not an ending but a beginning, the start of a shift that depends entirely on the stillness of others. The parrotfish solve this problem in a way that, described out of context, sounds implausible. As darkness falls, the animal positions itself in a cavity in the coral and begins to produce mucus from glands inside its gill chamber. Slowly, over the course of roughly an hour, the secretion builds outward from the body in all directions, forming a translucent envelope, a cocoon of mucus that entirely encases the fish. The primary function of this membrane is chemical concealment. The reef at night belongs to Nethiae isopods, tiny blood-feeding crustaceans that locate resting fish by following chemical trails through the water. The mucus barrier blocks that scent. Experiments conducted by Alexandra Grotter and her team at the University of Queensland in 2011 demonstrated the scale of the difference. Parrotfish that were prevented from forming the cocoon were attacked by isopods 95% of the time. Those that completed the envelope were attacked only 10% of the time. But the cocoon does more than mask odor. It functions as a tactile perimeter. If something contacts the outer surface of the membrane, a shifting current, the body of an approaching predator, the disturbance travels through the mucus and reaches the fish before the threat itself arrives. The animal wakes and bolts. An alarm system woven from the animal's own secretions, costing approximately 2.5% of its daily energy budget. A small expenditure for an unbroken night. Because the alternative is being located at night, inside the reef, by what comes out of the crevices after dark, has one outcome. The giant moray eel spends the daylight hours folded inside narrow fractures in the reef wall, its body coiled back on itself, jaws open, pumping water over its gills. It does not move. It does not hunt. It waits. When the light drains from the water column, the body begins to unfold. 3 meters of dense, flexible muscle, lacking the pectoral fins that other fish use for maneuvering, sliding along the reef face guided primarily by olfaction. The moray's eyes are small, poorly suited for detail. What drives the hunt is scent, chemical traces left by the animals hiding in the reef's interior. The moray's body is built for exactly the geometry of the reef at night. Narrow, cylindrical, capable of coiling through gaps and fissures no other predator of comparable size could enter. It pushes into cracks, uses its own body as leverage against the walls, and extracts fish that believed the coral itself would protect them. But the moray is not the only nocturnal predator working the reef. And in 2006, a team led by Ridwan Bishari observed something in the Red Sea that shifted what was understood about cognitive capacity in fish. A coral grouper, a large-bodied reef predator with excellent vision but too bulky to enter the reef's internal architecture, approached a resting moray and positioned itself directly in front of the eel's head. Then it shook its head, repeatedly, vigorously, an exaggerated, rhythmic lateral movement that had no function in locomotion or feeding. A signal. The moray emerged from its crevice and followed the grouper. The two hunted in coordination. The moray entered the narrow spaces within the coral, and the fish hiding inside panicked and fled into open water. The grouper, stationed outside, intercepted whatever escaped. When the moray failed to respond to the initial headshake, the grouper returned and repeated the gesture, insisting until the eel joined. 2 species of fish from different families with different anatomies and different hunting strategies, communicating through a learned visual signal and dividing labor based on complementary abilities. One flushes prey from shelter, the other captures what flees. Each animal must recognize that the gesture from the other carries meaning, that the meaning implies a specific cooperative action, and that both will benefit from the partnership. This is referential signaling. This is role-based coordination. Before Bishari's observations, both were considered exclusive to mammals. The reef at night is not simply a reef with the lights off, it is a separate ecosystem laid over the daytime one. A second architecture of behavior, occupying the same physical space but governed by an entirely different set of rules. Those who rest must vanish. Those who vanish must build defenses, and those who hunt must find every defense that fails. The darkness does not pause the reef, it rebuilds it from the inside out on a schedule that every organism reads and none can ignore. In the open ocean, far from any coastline or reef structure, darkness is not something that arrives on a schedule. It is a permanent condition. Below 200 meters, Sunlight ceases to exist. The water is black, cold, and featureless, a void that extends downward for thousands of meters without landmark or interruption. Most animals avoid this zone. The northern elephant seal descends into it deliberately because it is the only place where sleep is possible. Elephant seals spend months at sea on foraging migrations that cover thousands of kilometers. They do not haul out on rocks or beaches during these journeys. They do not rest at the surface. The surface is where the danger is. The open, sunlit layer of water where orcas patrol and great white sharks hunt from below, scanning for silhouettes against the light. An elephant seal floating motionless at the surface is a silhouette. And, unlike dolphins or whales, the elephant seal cannot partition its brain. It cannot rest one hemisphere while the other keeps watch. When this animal needs to sleep, the entire brain must go offline. Both hemispheres, full shutdown, which means the animal must find a place where being fully unconscious will not kill it. The solution is to fall. The dive begins under the animal's control. It descends in a smooth, directed glide, angling downward through the water column, past the last traces of blue-green light, past the thermocline, into total blackness. 200 meters, 300. The only sound is the faint drag of its own body against the water, and then even that fades as the descent slows. In the first minutes, the brain enters slow wave activity, the initial phase of deep rest, heavy and restorative. During this stage, the animal still maintains some postural control. The body stays oriented. Then the transition happens. The brain shifts into REM, rapid eye movement, the phase associated in mammals with dreaming, memory consolidation, and one critical physiological consequence, muscular paralysis. The voluntary muscles go slack. The flippers stop making corrections. The body loses all orientation. The elephant seal flips inverted and begins to spiral downward. Researchers at the Scripps Institution of Oceanography, led by Jessica Kendall-Barr, documented this behavior in 2023 using sensors that recorded brain activity, body position, and depth simultaneously in free-ranging animals at sea. The data showed a consistent pattern. As REM onset occurred, the animal's descent path shifted from a controlled glide to a corkscrew, a rotating, tightening helix aimed at the ocean floor. The initial loops were wide, roughly 7.5 meters in diameter. As the paralysis deepened and the sleep intensified, the spirals compressed to just over 3 meters across. The animal completed between 2 and 13 full rotations per episode, at depths ranging from 82 to nearly 400 meters below the surface. During these spirals, the elephant seal is entirely unconscious, paralyzed, inverted, spinning slowly through absolute darkness with no awareness of depth, orientation, or oxygen reserves. It does not know where it is. It cannot correct its trajectory. It cannot respond to a predator, a current shift, or the approaching sea floor. It is, in every measurable sense, absent. A body in freefall through a medium that offers no ground, no boundary, no signal that the descent must stop. This was the first confirmed recording of REM in a wild marine mammal in open ocean conditions, and the arithmetic governing it is ruthless. Each sleep episode lasts approximately 10 minutes per dive. The animal surfaces for 1 or 2 minutes to breathe, then descends again. Over an entire day at sea, the total accumulation of rest reaches roughly 2 hours. When the same animal hauls out on a beach during the breeding season, it sleeps 10 hours a day. The ocean compresses its rest budget by 80%. The calculation the body performs is precise and unforgiving. Descend below the hunting range of the apex predators that patrol the upper water column. Lose consciousness for 10 minutes in free fall. Wake before the oxygen runs out. Ascend, inhale, repeat. The margin between survival and death is measured in minutes, in meters, in the remaining partial pressure of oxygen in the blood. If the spiral carries the animal too deep, if the sea floor arrives before consciousness returns, if the reserves are insufficient to power the climb back to air, there is no waking up, no reflex to trigger an emergency ascent, no backup system. The brain is off, the muscles are locked. and the body continues to turn in the dark, indifferent to everything, including its own limits. In a laboratory at the Okinawa Institute of Science and Technology, an octopus is asleep inside a makeshift den. The arms are gathered beneath the body. The mantle is drawn in tight. The skin is pale, almost white, a uniform blank. Ultra-high-resolution cameras record everything. Every texture change, every contraction, every shift in pigmentation across the entire surface of the animal. For roughly 30 minutes, there is nothing to record. The body is still, the pulse is slow, the pupils are narrowed to thin slits. Responsiveness to external stimuli drops to almost nothing. This is quiet rest, a state of reduced activity that looks, by every behavioral measure, like the deep, slow wave phase that mammals enter at the beginning of a sleep cycle. Then, for approximately 60 seconds, the animal transforms, the skin erupts, brown patches bloom across the mantle, spread, dissolve. White bands streak along the arms, deep red surface, and vanish. The texture shifts, smooth skin turns granular, pebbled, rough, then flattens again in rolling waves. Beneath partially closed lids, the eyes move. The suckers along the arms grip and release in irregular pulses. The arms themselves twist slowly, involuntarily, as if reaching for something that is not there. The entire body becomes a display, vivid, rapid, complex, running through patterns that the animal uses during waking life. Camouflage sequences, threat displays, escape colorations. The repertoire of a conscious octopus replayed on the body of an unconscious one. This is active rest. In 2023, a team led by Aditi Pophel at the Okinawa Institute recorded the neural signatures accompanying these episodes and found that brain activity during the active phase closely resembled wakefulness. The regions most engaged were the superior frontal and vertical lobes, structures associated with learning and memory formation. The electrical profile was so similar to the waking state that distinguishing the two required careful reference to the behavioral context. The animal was not awake, but his brain was performing work that, in a waking animal, would correspond to processing experience, consolidating information, running through stored patterns. Which raises a question that the data can frame but not yet answer. If the brain during active rest is operating in a mode nearly identical to waking cognition, and the skin is simultaneously cycling through the chromatic and textual patterns that the animal deploys in conscious life, is the octopus replaying experience? Are the colours on the skin a readout of something happening inside? The chromatophores, the pigment-filled cells that produce the octopus's colour changes, are under direct neural control. Each pattern on the skin corresponds to a specific neural command. During waking life, those commands are issued in response to environmental input, a predator approaching, a rival displaying, a substrate to match. During active rest, the same commands fire without any external trigger. The display is generated from within. The neuroscientist Siddhartha Ribeiro, studying cephalopod cognition at the Brain Institute in Natal, observed that it is difficult to resist the impulse to interpret what is written on the skin of a resting octopus, but that science does not yet know what the animal experiences during those episodes. The patterns are there, the neural correlates are there. The subjective content, if any exists, remains inaccessible. What is accessible is the debt. When researchers deprived octopuses of rest, the animals compensated. On the next opportunity, they entered the active phase sooner and more frequently, as if the body were demanding repayment for what had been withheld. This phenomenon, rebound, is one of the defining signatures of true sleep across the animal kingdom. Mammals show it. Birds show it. The octopus shows it. The system insists on its due. And this is where the deeper strangeness lies. The octopus is an invertebrate. Its nervous system is organized along fundamentally different principles from any vertebrates. 2/3 of its neurons reside in the arms, not the central brain. Its lineage diverged from the vertebrate line more than 500 million years ago, before the evolution of bones, jaws, or any structure that would become a mammalian brain. There is no shared ancestor from which octopuses and mammals could have inherited a two-phase rest architecture. The resemblance between the octopus's quiet and active phases and the slow wave and REM stages of mammalian sleep is not a legacy. It is a reinvention. Evolution arrived at the same structural solution at least twice through completely independent pathways separated by 500 million years of divergence. When natural selection converges on an identical solution across lineages that share almost nothing in common, not anatomy, not neural architecture, not habitat, not lifespan, the solution is not a coincidence. It is a constraint. Something about the way nervous systems operate, whether centralized in a skull or distributed across 8 arms, requires periodic cycling between 2 distinct modes of reduced consciousness. The pressure that produces this pattern is older than the brain structures that execute it, and more fundamental than any single lineage's answer to it. The octopus did not inherit two-phase rest. It was compelled to build it from scratch, because the alternative, whatever the alternative is, does not work. On the surface of the North Pacific, there is no reef to hide in, no depth to escape to, no cave or crevice or sand to burrow under. The water is open, cold and exposed. Everything above it, wind, weather, predators scanning from below, can reach whatever floats there, and sea otters float there. They rest on their backs, belly up, paws folded on their chests or splayed to the sides, faces pointed at the sky. It is the most vulnerable posture imaginable for an animal in open ocean. No shelter, no concealment, no architecture between the body and whatever is looking for it. Unlike seals or cetaceans, sea otters carry no blubber. There is no thick subcutaneous fat layer insulating the body from water that, in the North Pacific, can hover just above freezing. What stands between the animal and hypothermia is fur, the densest fur of any mammal on Earth, approaching 1 million individual fibers per square inch. The coat traps a layer of air against the skin, and that air is what provides both thermal insulation and buoyancy. If the fur becomes soiled, matted, or coated with oil, the air layer collapses, insulation fails, buoyancy drops. Falling asleep under those conditions means drifting into cold water that the body can no longer resist, a process that ends in thermal collapse and drowning simultaneously. So the fur must stay clean, and the body must stay in place. To solve the second problem, sea otters wrap themselves in kelp. The long fronds of giant kelp grow from holdfasts on the sea floor all the way to the surface, sometimes spanning 30 meters or more of water column. An otter preparing to rest will roll itself into these fronds, winding the rubbery strands around its body until it is tethered to the canopy. The kelp acts as a mooring line, holding the animal at a fixed point while tides and currents move the water around it. The otter drifts within the radius the fronds allow, but does not travel. It stays. Where no kelp is available, the animals hold on to each other. Groups of otters resting together link paws across the water's surface, forming loose aggregations that move as a unit and prevent any single individual from being carried off by the current. These floating clusters are called rafts, and they can be enormous. Observations have recorded gatherings exceeding 2,000 animals, a sprawling mat of bodies bobbing in unison across the swell. The rafts organize themselves by sex. Males group with males, females and their young form separate assemblies. A mother who needs to dive for food will wrap her pup in kelp before descending, binding the infant to the canopy because the pup is too small to manage its own position in the water. Before she goes, she blows air into the pup's fur, inflating the coat to maximize warmth and flotation until she returns. At the surface, in full view, with nothing between rest and death, except the group, the kelp, and the integrity of the pelt. That is how sea otters manage unconsciousness. It is not a private act. It is a collective one, dependent on a living structure that the otters themselves maintain. Because the kelp forests are not just the anchor, they are the product. Sea otters are the primary predator of sea urchins along the Pacific coast. Urchins feed on kelp, grazing the holdfasts and stipes that keep the canopy connected to the bottom. Where otters are present, urchin populations remain controlled, and the forests stand. Where otters disappear, the urchins have no check. They multiply, they consume the kelp from the base upward, chewing through the holdfasts until entire forests detach and wash away. In the 18th and 19th centuries, the commercial fur trade reduced the North Pacific sea otter population from several 100,000 individuals to fewer than 2,000. The fur was too dense, too valuable, too perfectly suited to human use. As the otters vanished, the urchins exploded across the newly unguarded sea floor. The kelp forests collapsed. Vast underwater canopies that had anchored ecosystems, and anchored the otter's own rest, dissolved into barren rock flats carpeted with spines. Removing the otter from the ocean did not just eliminate a predator, it eliminated the structure that made the predator's own survival possible. The animal that held the kelp in place was the same animal that needed the kelp to hold it in place. Without one, the other ceased to function. The anchor depended on the thing it anchored, and the thing it anchored depended on it, and when the connection broke, both were lost. Every animal encountered in any study of sleep in the ocean, the whale that shuts down both hemispheres, the dolphin that alternates them, the shark that cannot stop moving, the seal that spirals unconscious through black water, the octopus whose skin replays waking patterns while it rests, possesses a brain. The brain may be divided, modified, partially deactivated, reduced to a minimal architecture, but it exists. There is a centralized organ that processes information, coordinates behavior, and, when the moment arrives, enters a measurably different state that the organism appears to require. Sleep, in every case studied up to this point, is something a brain does. Cassiopeia has no brain, no central nervous system, no concentrated mass of neural tissue, no structure that even loosely resembles the organ that, in every vertebrate and most complex invertebrates, is considered the seat of behavioral control. What Cassiopeia possesses is a diffuse nerve net, a decentralized web of neurons distributed across the body, with small clusters called rhopalia spaced around the margin of the bell. These clusters contain simple sensory structures, light-detecting cells, gravity-sensing crystals, but nothing that integrates information the way a brain does. There is no center, there is no hierarchy. The animal operates without anything that neuroscience would recognize as a command structure. Cassiopeia lives upside down. In the shallow, sun-warmed waters of mangrove flats and tropical lagoons, the jellyfish rests with its bell pressed against the sediment and its oral arms pointing upward, exposing the symbiotic algae embedded in its tissue to sunlight. It pulses rhythmically, slow, steady contractions of the bell that circulate water across its body, drawing in dissolved nutrients and microscopic food particles. The pulsing is constant during the day. It is the animal's primary behavior. Contract, relax, hour after hour, in water so shallow that the bottom is visible and the current barely moves. In 2017, a team at the California Institute of Technology decided to find out whether this animal, this organism without a brain, could meet the behavioral criteria for sleep. The setup was deliberately low-tech. The researchers positioned consumer start fabs above small aquaria containing individual Cassiopeia specimens, cameras facing downward, and let them record. No electroencephalogram was possible because there was no brain to attach electrodes to. No neural imaging, just video, shot from above, counting bell contractions over time. At night, when the laboratory lights went off, the pulsation rate dropped, not slightly, 32% below daytime levels. The animals were not just slowing in response to darkness, they were entering a state with measurably different characteristics. Reaction times increased, when the researchers introduced a stimulus, lifting the platform the jellyfish rested on. The animals took longer to respond than they did during the day. The body had shifted into a mode that reduced both output and sensitivity. Then came the critical test. The team began disrupting the nighttime state, firing pulses of water at the animals every 20 minutes throughout the night, preventing them from settling into the low-activity phase. The next day, the Cassiopeia that had been kept active overnight were 17% less active than normal. They pulsed less. They responded more sluggishly. They behaved in every measurable way, as if they were compensating for what had been taken from them. The body was collecting on a debt. 3 behavioral criteria define sleep across the animal kingdom, regardless of species. First, a period of reduced activity and responsiveness. Second, a rapid return to full alertness when stimulated. The state is reversible, not pathological. Third, a rebound effect when the state is prevented. The organism compensates afterward, as though the need accumulates. Cassiopeia satisfied all 3, without a single centralized synapse. The finding, published in Current Biology, represented the first documented case of sleep behavior in an organism entirely lacking a brain. In 2025, researchers at the University of California, Berkeley, pushed the discovery further. Examining the repalia, the small marginal ganglia distributed around the bell's edge, they identified cholinergic receptors involved in regulating the transition between the active and quiescent states. Cholinergic signaling, the same molecular system that governs arousal and rest in vertebrates, from fish to humans. Not a similar system, the same neurotransmitter pathway, conserved across a divide that spans more than 600 million years of evolutionary separation. The machinery that produces sleep is older than the organ we assumed was required to generate it. It predates centralized nervous systems. It predates bilateral symmetry. It predates the existence of anything that could be called a thought. Whatever sleep does, whatever function it serves that is so essential that evolution preserved it across every known lineage, in every known habitat, in bodies with and without spines, shells, skulls, or brains, that function was already operating before the architecture of consciousness had been assembled. The need to rest came first. The capacity to think came after.